Welcome to this biochemistry lecture on the central dogma. It's commonly referred to as central dogma of biology, central dogma of molecular biology, and it can be summed up very succinctly by the statement DNA makes RNA that makes protein. And you can see that in this diagram where we have DNA that's transcribed to make RNA, and then we have RNA that is translated to make protein. Those processes that are associated with this are transcription, translation, and replication, which occurs when a cell divides and you need an extra copy of the DNA. That's the central dogma of molecular biology. And there's associated DNA structures. We'll talk about that in a different unit. Today, we're going to focus primarily on transcription, translation, and replication in order to understand basically the important parts of biology. First, transcription. Transcription is taking and going from DNA to RNA. Very, very straightforward. Over here you can see we're going to take our DNA, our genomic DNA, and we're going to make RNA. This happens in a process where the RNA polymerase, actually what ends up happening is you have, and we'll go into greater detail on this in a different lecture, but you have, on the promoter region, you have activators, co-activators, DNA bending proteins, repressors, and enhancers that are going to bind. At the initiation site, you have RNA polymerase that's going to open up the DNA. And that opening of the DNA is going to then allow the polymerase to make a copy of that DNA made out of RNA. So there is hybridization that goes on. And the end product of all of this is an RNA transcript, an mRNA transcript, a messenger RNA transcript that is then, in the case of eukaryotes, moved. It's going to go out into the nucleus where it's going to be translated. In the case of prokaryotes, of bacteria, and simple cell organisms, translation can happen right there or nearby, because there's no real nucleus in a prokaryote, so it happens in the cytosol. So that's transcription. Transcription, again, we take and we go from DNA to RNA. Again, we'll go into greater detail on this as we get through. The entire process of transcribing and translation is known as gene expression. So we just talked about transcription. Gene expression is taking the mRNA and making it into protein. And this is an EM, electron microscopy image of translation occurring on mRNA. So you have these long fibers. You can see the green arrow where this is an mRNA transcript, and then these hair-like structures are all ribosomes that come in, and the proteins are being translated off that mRNA. So the entire process, again, gene expression, transcription, and translation occurs, and it can occur simultaneously. And I think the important part is that one mRNA transcript can have multiple amino acid transcripts that are built from it. Again, this is translation. Translation is going from RNA, the mRNA transcript, to protein. The transcription happens up top, and that happens in the nucleus in eukaryotes. And again, we have translation that occurs in the cytoplasm, and this is the major enzyme associated with translation is the ribosome, which is composed of a large subunit and a small subunit. We have this transcript, this mRNA transcript, that's going to be transcribed, and we have a growing polypeptide chain that's going to grow off that. So polypeptides are just floating around in the cell. They're going to attach to a tRNA, which we have right here. Over here, those tRNAs are going to come in and bind into this ribosomal subunit, and you're going to have a chemical reaction where the peptide bond is formed, and as it goes through, you're going to make a growing polypeptide chain. Translation. 3 steps in translation: initiation, elongation, termination. Should be self-explanatory. Initiation is where the ribosome subunit forms onto the mRNA transcript. Elongation is where we add all of these amino acids into a long string, and then the termination is when the stop codon is hit and From there, the entire ribosome pops off, and then it can go back and do it all over again. That's why you see so many transcripts in that EM on one subunit. Again, we'll go over all of this in greater detail as we get going. The last element of the central dogma is replication. Replication is when two cells, or one cell is dividing into two cells, and we need to make two copies of the DNA. So the DNA opens up. We make copies on both strands, on the leading strand and the lagging strand. And from there, we end up with two copies. So there's a polymerase, a DNA polymerase, and a what's called a replication fork that occurs as we open up this thing and open up the DNA. And from there, we're going to keep adding nucleotides that are complementary to the other strand. This allows for an exact copy of your DNA, the entire thing, to be made. And we'll talk about this in greater detail, but the replication aspect of DNA is when we need to make an exact copy of the genomic DNA in order to have an extra copy for cell division. So that's replication. Again, going back to the purpose of this video, central dogma. Central dogma of molecular biology is DNA goes to RNA, goes to protein. And that's primarily the topic in a nutshell. We'll go into each one of those topics in greater detail, and they're in an individual section and on the individual slideshow, but that's the important part about what we're talking about today. Look forward to talking to you later.